Start this recording. And I'll minimize this for you. You have your laser pointer. It's a full 15 minutes.
Okay.
So today, we have Dr. Lawrenz from downtown. That's the medicine, the microbiology. He works with them. UofL cancer for in here today to talk to you. Anything you want to know about?
Well, I appreciate the invitation to come over here. I actually presented in bioengineering 5 or 6 years ago. And so it's good to come back and give another talk. And, you know, we're over on the health science campus, and the Center for Predictive Medicine is really focused on biodefense and emerging infectious diseases. There's a lot of researchers over there that are interested in drug development, drug delivery. We have bioengineering faculty as part of that center. We have faculty from a lot of different places. A lot of people interested in vaccine development. I only bring this up because there are probably opportunities over there for bioengineering students. And so if you guys are interested in infectious disease and drug development, you should always take a look over there if you're looking for places to do some research. As Nick said, I'm going to talk to you guys about the plague. This is, of course, a human disease that's famous historically for a bunch of pandemics that have occurred. It's estimated that about 100 million plus people died from the plague during those 3 pandemics, including the Black Death. This is a disease that's caused by a bacterium known as Yersinia pestis, and it's primarily a zoonotic. So that means that it's not really evolved to be a human pathogen, but it lives in wild populations around the world. It's endemic across the globe, including here in the United States. So what I'm showing you on this map here is that every single one of these green states are areas that we have evidence that plague is present in. So it's endemic in the Western United States. It's endemic in these zoonotic cycles between small rodents and the vector that transmits this disease, which are fleas. And you can see that this is confined to the Western United States. And that's because this is an old world disease. It only came into the US in March of 1900. There's some beautiful epidemiology on this during the 3rd plague pandemic. They know which boat it came on, the port of San Francisco. 3 months later, there was another boat that came into Seattle. And what we have been seeing is the organism's been spreading eastward ever since. Now, as a zoonotic infection, where we see spillover events into the human population, then, of course, it's going to occur in areas where the bacteria is present in the wildlife population. So all of the yellow dots on this represent human plague cases over the last 100 years. You may have seen we had about 4 cases 2 months ago that hit the news really big, including the individual who died from pneumonic plague in Arizona. And so we do see this in the US, and it is moving eastward. We don't have it here in Kentucky yet, but there's no reason to think that it's not going to move this direction. And climate change is a big deal here, right? For all zoonotic pathogens, for all vector-borne diseases, changes in climate where we can change the habitat or have longer summers where the insects thrive are going to result in more spillover events. Now, the disease and manifestations that we see when we have that spillover into humans can manifest in 3 different forms. So the most common form of plague is bubonic plague. This is what happens when you are bitten by an infected flea. The bacteria sets up shop in the dermis, but really quickly enters into the draining lymphatics and will go to the draining lymph node. Now, a lymph node is typically not a place that bacteria like to find themselves, or we mount immune responses against infection. But Yersinia does a really good job of hiding from the immune system in the lymph nodes. They grow to really high concentration, and these lymph nodes will get really swollen and tender. So they can get about the size of my fist. So if you can imagine a lymph node under your armpit about that big. And these are typically referred to as buboes, and that's where the term bubonic plague comes from. Now, if an individual doesn't receive treatment for bubonic plague, the organism can disseminate into the bloodstream. And in the natural course of the disease, this is the ultimate goal of the bacteria. Getting into the bloodstream allows it to get to a new flea, that allows it to get to a new host. But from a clinical perspective, where we get really concerned about septicemic plague is the fact that in the bloodstream, now it can move to other tissues. And clinicians are really concerned about this when it gets into the lung and you get the development of pneumonic plague. And that's because once it gets into the lungs, it can be aerosolized. And now you can see person-to-person transmission. You don't need that flea anymore, and you get person-to-person pneumonic plague. So as I said, we have a few cases, about 5 or 6 cases annually in the US. There are other places that we see a lot more cases. One example of this is Madagascar. So Madagascar is an island off the coast of Africa. It's endemic for plague. And typically we see about 500 cases of plague, human plague in Madagascar on an annual basis. In 2017, there was a big epidemic of plague. So you can see on this graph, this is just showing the incidence of plague over about a 4-month period, I think. But there were almost 2,400 cases of plague during that period. So 500 or 5-fold more, I sound like Trump, 500 times more, 5-fold more cases in that 4-month period than what they typically see annually. And the red bars there highlight cases of pneumonic plague. What happened here was that we got a case of bubonic plague in the capital city that then triggered a pneumonic plague outbreak and that person-to-person transmission. There was a lot of scare that someone was going to get on a plane, flying with the plague, fly to mainland Africa and cause an outbreak beyond the borders of Madagascar. Luckily, the World Health Organization and the Institut Pasteur were able to contain this infection, and it ramped down by the end of the year. The other thing that I want to highlight here is those days that I have up there in red. That's how long it takes for someone untreated to die from this disease. So you can see, depending on the route of transmission, people can succumb to this infection in as little as 3 days post-exposure. Now, what this means from an evolutionary standpoint is that all the pressure that's put on this bacteria in the mammalian host is really driven by the innate immune response. In this short period of time, we don't have the ability for the adaptive response to respond that quickly. It's important, but in this case, all the bacteria cares about is what's going on with the innate immune system. And so it's evolved a wide variety of virulent mechanisms that allow it to really cope with these first responders to infection, the white blood cells that we know as the macrophages. So because of this bacteria actually turns out to be a really powerful tool for us to probe and better understand the innate immune response, to look at the ways that it's able to evade clearance by these cells, gives us insight into how these cells normally kill bacteria and other pathogens. And so we've really applied that in our lab to understand this dynamics between the host immune response and the bacterial pathogen. Now, if you look at the research that's going on in my lab, it actually falls into a couple buckets. So about half of my lab is really interested in those direct interactions with those myeloid cells, the macrophages and the neutrophils. I'm showing you a couple videos up here. One of these on the top is a macrophage that's infected with Yersinia pestis. It's in red. I hope you can see that it is definitely winning that battle with the macrophage. It gets inside, but the macrophage doesn't kill it. But more recently, we've been really interested in understanding how neutrophils, which are on the bottom, you can see them chasing the bacteria down there, are unable to control this infection when they interact with it. The other half of my lab is really interested in this phenomenon known as nutritional immunity. When I came about 5 years ago or 6 years ago, I talked about the work that we were doing in the nutritional immunity side of stuff. Nutritional immunity is a very innate response that we have that restricts nutrients from invading pathogens. One of the nutrients that we really restrict are metals so like iron, zinc, manganese. And in order for any pathogen to colonize you and I as a host, they have to figure out ways to get these metals because all life on the planet requires metals in order to live. And so we're really interested in how the bacteria is able to do that and overcome nutritional immunity during infection. Now, ultimately, if you look at this, and what I'm going to talk to you guys about today is really basic science research. We want to understand the pathogenesis of the organism. We want to understand how you and I respond to infection. But what we hope to be able to do is as we understand these processes, we can take those and then translationally apply these to new therapeutics. And these could be therapeutics for plague, but really we imagine these as being broader to go against other bacterial pathogens. So there's a lot of focus in some of the research going on in my lab in the development of new vaccines, and we work with a lot of people on adjuvant development. We have a really good vaccine model for this. But as we understand how Yersinia is able to subvert the immune response, we also want to apply those towards immunomodulation. So for example, ways that we can put molecules in nanoparticles and treat macrophages and then have the macrophage win that battle up top, not the red bacteria. So with that in mind, what I want to talk to you guys about today, I'm going to tell you two short stories about how Yersinia pestis subverts or manipulates these neutrophils in order to cause infection. And I'm going to tell you two stories by two very talented graduate students in my lab. Amanda Brady, who graduated last year and is now a postdoc at the University of Colorado. And then Caitlin Shinneman, who's the current graduate student in the lab, who's hoping to graduate next year. So I'm going to start with Amanda's work. And to get into this, I want to introduce you guys to one of the models that we use to study plague. And so this is the course of pneumonic plague. There's a simple representation of what goes on in the host during pneumonic plague. So you can see here, this is data from a mouse model, but it's exactly the same as what happens in humans. When the bacteria gains entry into the host, it'll replicate almost logarithmically for about 72 hours. And then if it goes untreated, the human or the mouse will succumb to it. The interesting part about this is that we see that it's interacting with the innate immune system during this period of time. So we have tricks to look at what cells are directly interacting with the bacteria, and I can talk about those afterwards if you're interested. But we know during the first 6 hours that the alveolar macrophages, the sentinel macrophages in the lungs, are the first cells that are interacting with the bacteria. But by 12 hours, neutrophils become the primary cells that are interacting with the bacteria. So we see these interactions with the first responders, but the amazing part is we don't see that the host is really mounting an inflammatory response. And so if we look at the inflammatory response, we see that this is significantly delayed in pneumonic plague as opposed to other pulmonary infections with gram-negative bacteria. So I'm kind of showing that in the blue line up here as a representation of inflammation. So this is another way to look at this. So this is histology of the lung. I am not a pathologist. Yep. They are the first responders. So what's happening here is we're getting the resident neutrophils interacting. We're not pulling them from the bloodstream like we normally would. And that's highlighted here because at 24 hours, when we would normally expect there to be a lot of neutrophils coming in, there's not. So if you don't look at lungs, you can be a pathologist of lungs. It's really easy to look at. If you have open alveolar air spaces with nothing in them, you can breathe. If you get neutrophils coming in, you get the development of pneumonia, you can no longer breathe. You don't have those open air spaces. And so what we see, even though there's interactions with neutrophils in 24 hours, we're not recruiting those neutrophils in. So we kind of refer to this as a biphasic immune response. And so there's this non-inflammatory phase for about the first 36 hours, and then a significant shift between 36 and 48 to this robust pro-inflammatory response that results in pneumonia because all these neutrophils are coming in, and that leads to the pathology that's associated with death. Now, what I can tell you is that that length of that non-inflammatory phase is really unique for pneumonic plague. And on top of that, we know that if we can artificially induce inflammation earlier, we can protect the animals from the death. So we can do this by putting cytokines directly into the lungs, recruiting in neutrophils earlier, and you'll see that this results in earlier inflammation, control of bacterial growth, and more importantly, we can go from almost all the animals dying, 60% of them surviving infection. So the development of that non-inflammatory phase is really important for the establishment of lethal disease and infection by Yersinia pestis. And because of this, really one of the central questions in my laboratory in the Yersinia field in general is how does this organism inhibit the recruitment of those neutrophils? Or how does it generate that non-inflammatory environment that appears to be essential for it to cause disease? Now, we're going to go back and we're going to talk about inflammation. So I'll give you a little bit of review on this. Bringing neutrophils into the lungs when there isn't an infection is a bad idea, right? They come in, you can't breathe, you get tissue damage, etc., right? So controlling inflammation is a really tightly regulated sequence of events. We only want to bring those neutrophils in when there's actually an infection that we want to fight. And so to do this, what we see are a couple different phases of the inflammatory phase. So I break this down into two. The first phase that I look at is called initiation, and that's driven by the sentinel leukocytes in the lungs. That's going to be the alveolar macrophages. Those are the first cells that are going to interact with the bacteria. And what they do during that first initial interaction is they develop lipid mediators of inflammation really quickly. And those lipid mediators will recruit resident macrophages and neutrophils to the site of infection, so within the tissue and bringing them to the sites of infection. Those resident cells will make additional lipid mediators, but then they'll start to make protein mediators that we're more familiar with, the cytokines and the chemokines that are needed to amplify this system and actually start triggering those circulating neutrophils to come into the tissue. Now, we've known for a long time, I showed you a little bit of data, that there's a delay in the inflammation of the neutrophils. But this correlates very well with the delay in that amplification step. We don't see the production of those cytokines and chemokines that are needed to bring those neutrophils up. But surprisingly, even though initiation is absolutely required for starting this inflammatory cascade and getting a timely response, nobody had really looked at what was going on in those early interactions between Yersinia pestis and those sentinel macrophages and neutrophils. And that was until Amanda joined the lab, and she really focused on that initiation step and published two really exciting papers understanding the first things that were going on during the initiation step. I'm not going to go into that data, but we'll talk a little bit about where she went on those studies. The important part is that she identified that this very important inflammatory mediator called leukotriene B4 was not being produced during infection. So LTB4 is, as I said, a lipid mediator. This is the structure of it up here. It's an icosanoid. It's synthesized by mast cells, neutrophils, and macrophages, so only by your leukocytes. And it's synthesized by this lipoxygenase pathway. Because it's a lipid, it can be produced really quickly. It doesn't have to go through gene transcription and translation. So that's why it's one of these things that initiates the inflammatory cascade. It initiates the cascade by interacting with a high affinity receptor on all of our immune cells called CLP1. And it is probably the most potent chemoattractant that you and I make. And more important than that, it's the chemoattractant that's required for this phenomenon known as neutrophil swarming. So this is a neutrophil swarm here, and you can see while they might migrate to the tissue, where that tissue damage is, you see those neutrophils swarm in as a big group around that. And that can help control infection or look at regulate tissue damage response. If you remove LTB4 or BFP1, you won't see those swarms. They might come into the area, but you don't get that concerted event on very tight locations. In addition to being a chemoattractant, this can also prime cells to better respond to infection. So this again is from Amanda's work, where she took macrophages and just treated them with LTB4 and then fed them Yersinia pestis. You can see normally Yersinia pestis wins that battle. I showed you a video of that. But if we prime these with LTB4, they can become better able at killing the bacteria. And there's been evidence in the literature for a long time that LTB4 is important to initiate the inflammatory cascade to control bacterial infections, fungal infections, parasitic infections, viral infections, you name it. And the reason, again, that Amanda got really excited about this is that when we looked in the host, we really didn't recover much LTB4 during that non-inflammatory phase. We only saw it in that pro-inflammatory phase later during infection. So indicating that perhaps the organism wasn't initiating this cascade during infection. So she wanted to look at what happens when this organism interacts with leukocytes. So we did a reductionist approach here now. We moved out of the animal host. We moved into cell culture. I'm going to show you a lot of work on neutrophils. Again, we focus on neutrophils because they're the primary cells that are interacting, but you'll see along the way, I'll show you some macrophages and other cell types too. First thing that Amanda wanted to do is that we were working with neutrophils in vitro, could she actually stimulate them to make LTB4 in response to it? So the first graph up here is just showing you LTB4 production by neutrophils stimulated with a wide variety of different gram-negative bacteria. So E. coli, Salmonella, Klebsiella. You can see that you get this production with LTB4, you get a lot against Klebsiella. But the amazing thing was, if she stimulated these neutrophils with Yersinia pestis, absolutely no LTB4. And in fact, if she put 100 bacteria per neutrophil, she still saw no LTB4. So it didn't look like these neutrophils were even recognizing that the bacteria were there. This was not conserved just in neutrophils. If she went to the other cells that can make LTB4, macrophages or mast cells, she saw that they didn't produce any LTB4 in response to this bacterium. Now, one question that I get a lot is the bacteria just killing the cells? And eventually, Yersinia will kill phagocytic cells, but it takes a while. We're looking at 1 hour post-infection. And at 1 hour post-infection, these cells appear to be viable. I love where we will get there. I love where you're going. Amanda had the same question. So the ultimate goal here is, why is the neutrophil not recognized? We'll get there. But I want to highlight that they're not dead. So they're just as viable if bacteria are there or not. And they're metabolically active. If you hit them with ionomycin, which we know stimulates LTB4, it's just a chemical that causes change in calcium flux, you can see that you can make as much LTB4 as an unaffected cell. So these cells aren't dead. There must be something else going on, hiding from the neutrophil or manipulating the neutrophil. All right. So at this point, I'm going to move away from the immune system. I'm going to tell you a little bit about the biology of the bacteria. So this type 3 secretion system is the primary virulence factor for Yersinia. And if you're not familiar with type 3 secretion system, you can think of these as the bacteria bioengineering a syringe that it can use to move bacterial protein from the bacteria directly into the host cell. And so in the case of Yersinia, what it does is it has 7 proteins that go through the system and get injected into the host cell. They target host protein, disrupt host signaling, and the outcome of that is that this system is required to cause disease. You remove the type 3 secretion system, it's attenuated. We work with it on the bench in the laboratory. But more importantly, we know that this system is absolutely required to generate that non-inflammatory phase of the biphasic immune response that we talked about. And so Amanda proposed the hypothesis that perhaps the reason that we're not seeing LTB4 synthesis in response to this bacterium is because it's using this type 3 secretion system to manipulate the cell. So to test this, what she did was generate a mutant that was lacking all of the proteins that move through this system. And now you can see for the first time, I'll refer to this as the T3E or the effectorless mutant. For the first time, we're getting LTB4 synthesis by neutrophils in response to pestis. So really indicating that those proteins, which are called YAP effectors, are manipulating the cell and blocking the production. This is an active process. And she showed this by doing co-infection. So she infected neutrophils with Klebsiella, who makes a lot of LTB4. I showed you that earlier. But she co-infected those now with Yersinia, and you can see it comes down. So it can even block the production of LTB4 that would normally be made in response to Klebsiella. She went on to show that there are 4 of those proteins that get secreted through, down here highlighted in yellow, that are sufficient to block LTB4. This is redundancy. When we see redundancy in bacterial pathogens, it usually means it's pretty important for the pathogenicity and highlights the importance of blocking this system. So it potentially wasn't that surprising that the type 3 secretion system is manipulating the cell to block LTB4 synthesis. But for me, where this got really exciting is when Amanda, being a good PhD student, ran off and did an experiment without talking to me beforehand about it. Because I don't know that I would have had her do this experiment. But what she did is she infected neutrophils with a different strain of Yersinia pestis. And what she did was infect it with a strain that not only lacked those proteins that go into the host cell, but the needle itself. And this was really crazy to us. If you remove the needle, that's this one right here, the T3, now the neutrophils no longer made LTB4, suggesting that the neutrophils actually recognize the needle that the bacteria uses to intoxicate the host cell as a PAM to trigger the production of LTB4. And so in the case of Yersinia, that needle gets sensed, but then it puts proteins through to block it. Turns out that really what it's sensing is probably the pore that gets put into the plasma membrane of the host cell. So she could just remove the proteins that go into the plasma membrane and it no longer made LTB4. This isn't just in neutrophils. We see this in macrophages also, that it recognizes that needle. And this isn't just Yersinia. If we took other strains of bacteria that make type 3 secretion systems, I'm showing Salmonella up here, we see the same phenomenon. You don't get the production of LTB4 in response to strains that don't have that type 3 secretion system. So normally, if I was in a microbiology and immunology department, I quiz you guys on how this is done. I'll jump past that today, and I'll tell you that the type 3 secretion system we know is a pattern that is recognized by the immune cell. And what gets recognized, it gets recognized by the inflammasome system. And so the inflammasome system is a way for cells to recognize intracellular pathogens and then initiate a pro-inflammatory form of cell death known as pyroptosis. So what happens here is we know that the pore in the membrane is recognized by NLRP3. This causes production of the inflammasome, leading to cleavage of pro-inflammatory cytokines and then this pro-inflammatory cell death. But people have never looked at whether or not pro-inflammatory lipids were induced in response to the inflammasome. And so that's what Amanda wanted to do. And so what she did is we got mouse models in which they either don't make NLRP3 or they don't make caspase-1, so they're defective in that inflammasome pathway. She infected macrophages with that. That's shown here, and you can see they no longer make LTB4, indicating that the inflammasome pathway is what's recognizing the needle and triggering the production of LTB4. The real exciting part here is that if we go into neutrophils and do this, actually, it's not dependent on inflammasome activation. So you still see production of LTB4 in neutrophils, even though those inflammasome compartments aren't there. So some other system is recognizing and leading to the production of LTB4 in neutrophils. So as I said, we like to use pestis as a tool to probe the immune system. And this is where we can really leverage our understanding of the biology of Yersinia pestis to get at this question. So I told you earlier that there are a group of effectors that block LTB4 in the and one of those is YAPH. So what Amanda can do is she can go in infect with just YAPH, it doesn't have any of the other effector proteins, and it blocks the production of LTB4. Now we know from work from Joan Mexis's group that YAPH specifically blocks a signaling pathway in the host cell targeting SCAP2 that ultimately blocks calcium flux in the cell. And I didn't tell you this, but LTB4 synthesis, one of the signals that it requires is calcium flux. The enzymes need to bind to calcium to become active, and so that's required to get LTB4. So based on our understanding of the YAPH and the YAPH mutant, she hypothesized that this signaling pathway was involved in the neutrophils in leading to LTB4 synthesis. And so we got SCAP2 knockout mice, we got neutrophils from them, and you can see now that it no longer produces LTB4. So in neutrophils, it appears to be dependent on this SCAP2 PLC signaling pathway. And what was really refreshing was when we went into macrophages, which we knew were dependent on different signaling pathways, SCAP2 was not dependent. I'm not showing you the data, but YAPH on its own doesn't block LTB4 synthesis in macrophages. So this made a lot of sense to us. So I'm going to show you a model here of what Amanda came up with. What she really discovered was this novel recognition of the type 3 secretion system by leukocytes leading to the production of LTB4. In neutrophils, this goes through the SCAP2 pathway, but in macrophages, it's very different and goes through the NLRP3 and inflammasome pathway. Now, we're still actively working on this. In neutrophils, we're working our way upstream. Ultimately, we want to find this unknown, which is the receptor that's recognizing that complex in the plasma membrane. In macrophages, we still know that calcium flux is important, and it occurs through STIM1, but not through PLC. It's independent of PLC. And this is really novel. Typically, STIM1 requires PLC. This is the first evidence that we've seen in the literature that you can get STIM1 activation independent of PLC. All right, I'm going to spend about 10 minutes, that should get us to the end of the session here, telling you about Caitlin's work. So Caitlin's also interested in neutrophils. She's interested in this ability to hide from neutrophils. But instead of looking at lipids, Caitlin's really interested in extracellular vesicles. So if you're not familiar with extracellular vesicles, these are essentially liposomes that are made by all of our cells. They're produced at all times by our cells and released into the environment. And these extracellular vesicles are composed not only of lipids, but also other components from the cell. Host proteins, you can see small RNAs. For something like a neutrophil that can eat bacteria, you can get bacterial proteins packaged into it. But the important part about EVs is that the cargo that goes into these EVs are dependent on the physiological environment of the cell. So they will change depending on where the cell is or changes in physiology of that cell. So for something like a neutrophil, the biggest change that it's going to go through is it's going to find a pathogen and it's going to start to change the cargo that gets packaged into it. And that cargo really has implications on how these EVs can function in intercellular communication. These have been studied a wide variety of cancer research, but in inflammation, they really are starting to be seen as important signals to your bystanders to prime them locally in your tissue. But because these are lipid, essentially liposomes, they're really protected against enzymes and proteases, et cetera. And so they can travel with that cargo a long distance. So for example, they can get into lymphatics, go to the lymph node, and prime cells very far away, telling them there's something going on in the tissue, maybe we need to be prepared for an infection. Now for neutrophils, these EVs can have other functions besides intercellular communication. You can package antimicrobials directly into EVs, so EVs go out and kill pathogens directly by interacting. Or they can act as decoys. So this was shown really elegantly by Ken Cadwell's group, where he showed that cells will package the H2 receptor into EVs. That's the receptor for SARS-CoV-2, of course, and the virus would then target the EVs instead of the host cell itself. He also showed that neutrophils will package the receptor for leukotoxins, which are made by Staph aureus. This results in the toxin targeting the EV and not the host cell itself. Now, because of the role of these EVs in intracellular communication and priming the immune response to effectively respond to infection, Caitlin, when she joined the lab, had a really simple question, and that was, is the EV response by these neutrophils disrupted by Yersinia pestis? And so the first thing she did was she went in vivo again, like Amanda. I'll remind you of that 5-phasic immune response. And what Caitlin wanted to know is, does the EV production change depending on the phase of that immune response? So she harvested samples at 0, which are uninfected, 24 hours, which is that non-inflammatory response, or 48 hours, which is that pro-inflammatory response. She isolated EVs by differential centrifugation and then characterized these EVs. And so the first thing that she saw was that during the early stages of infection, there really didn't appear to be much in the change of the number of EVs within the lung environment. However, when we hit that pro-inflammatory phase at 48 hours, you can now see a significant increase in the number of EVs that we recover from the lung. This correlates directly with protein concentration, as we would expect. And when she went in by mass spec and looked at what proteins were in those EVs, what you can see is that there's a change in the cargo over time. You get a change at 24, and then you get a significant change at 48 hours. So again, like Amanda, Caitlin wanted to take a reductionist approach now, go into cell culture, and see if the bacteria is directly manipulating the neutrophil to change the cargo or the number of EVs that are. So in this case, we're doing human neutrophils here, and we're infecting those again with our 2 strains of Yersinia, the wild type strain that has all of the Yop effector protein, and then that T3E strain to test the hypothesis that maybe intoxication with the Yop effectors alters EV expression. This is a 1-hour infection. She isolates the EVs by differential centrifugation, and then she characterizes them all over again. This is our first view of, if you haven't seen an EV before, this is electron microscopy looking at EVs. They're typically anywhere from 100 to 400 nanometers, so they're very small. They get this kind of donut-like shape when they get fixed. But again, it's these vesicles that have this cargo in it. We see that when we challenge neutrophils with either of the bacteria, the size, average size changes. And this is different than if we challenge them with zymosin, which is a component of a fungal cell wall that we know stimulates EV production. when we look at how many EVs are being produced by the cells, the first thing that she saw was that if she stimulates them with wild type Yersinia, see no difference in the number of particles that are being produced compared to uninfected. However, if we remove those YAP effectors, those proteins that get injected into the host cell, we now see a significant increase in the number of EVs correlating with protein concentration as we expect. We did a lot of work to make sure that we had clean EVs here. I'm showing you some markers for EVs that are known to be cargo in EVs, but more importantly, we showed that there was exclusion of non-EV contaminants. And this gave us a lot of confidence that if we go in and look at the proteins that are in here, that these are proteins that are inside of EVs and not just garbage proteins that are been released by the cells. And so we did mass spec again. I'm sorry, hopefully you guys are familiar with the PCA, but this is really just a way to look at proteins or anything and compare groups. And if they start to separate, you can actually do statistics on this too. You can start to argue that this guy is different than this guy is different than that. Sometimes, being a biologist, I like to look at things a different way. So this is just a heat map of the top 50 proteins that are differentially enriched in our samples. There are about 300 total that are differentially, but I'm only showing you the top 50 here. Doesn't matter what these proteins are. What I want to highlight is if you look at the uninfected group, I'm hoping it's really easy by color that you guys can see that that group looks very different than any of the EVs from the bacterial stimulated. And then if you focus on the effector list, you can see this group down here that is highly enriched in the effector list that isn't in the wild type. And I know you guys can't see what's printed there. You'll take my word for it or go to the paper if you're interested in it. But that group that's right there, a lot of those are antimicrobial components that are made by neutrophils that are being packaged in these. So we see this difference in cargo, but really the question is, does that have any implication on the function of these, right? And that's what Caitlin wanted to do next, is ask, how do these alterations maybe alter the function? So I'll remind you, there are different functions that EVs can have, and we focus on two of these. The first one that we focused on was the antimicrobial. That was because we saw all of those antimicrobial peptides in the EVs themselves. But then we also want to look at that ability to communicate with those bystander cells and maybe tell them that there's an infection going on, even though they haven't directly interacted with the bacteria. So it's a really simple assay to look at antimicrobial. We take the bacteria, we incubate them with EVs, we let it sit for 4 hours, and then we just plate the bacteria on agar and ask how many bacteria are there. So you can see EVs from uninfected, we see no difference than untreated bacteria 4 hours later. That's true for the Y pestis EVs, but the EVs from those neutrophils that were stimulated with the effectorless mutants that had more of those antimicrobials were able to knock down 90% of the bacteria in that culture. Now we wanted to look at this intracellular communication. So to do this, what Amanda did, Caitlin did, was take naive macrophages and treat them with these EVs. And so macrophages come in different flavors, right? So typically you have naive macrophages, and macrophages can do a lot of different things. They can fight infection, or they can be involved in tissue remodeling and resolution of infection and repair. Typically, those that are going to fight infection will polarize to an M1 phenotype. So M1, bad for bacteria. M2 is the phenotype where they're going in and cleaning up and repairing tissue. That's M2. M2 is good for bacteria because they don't have all those antimicrobials. So what we asked was, if we treated these naive macrophages with these EVs, did it change the polarization? So if you treat them with interferon gamma, that's known to stimulate an M1 phenotype, a pro-inflammatory phenotype. You can see that that dramatically drives us towards an M1 phenotype. We treat those with the effectorless mutants, they look like interferon gamma. M1 pro-inflammatory. But the wild type bacteria don't stimulate EVs that lead to the ability to prime neutrophils towards that M1. And where this is really exciting is that if we take those macrophages and feed them Yersinia pestis, you can see that EVs from the wild type those macrophages aren't very good at killing the bacteria. In fact, they might have a slight advantage compared to untreated macrophages. But those from the effectorless mutants are really able to kill the bacteria on the level of priming these macrophages with interferon. So we have a bit of a model here. I'm going to show you what it is, and then if you guys have questions, I'm happy to answer any. Normally during infection, what you see is that neutrophils are going to interact with that pathogen. That's going to cause a signaling cascade that's going to lead to changes in the cargo that gets packaged in the EV. They'll change how many EVs get released by the host cell. And those EVs that are released are carrying things that can directly kill bacteria or stimulate bystander cells to be ready to better respond to that infection. However, when we look at this in the context of Yersinia, during those early interactions through that type 3 secretion system, it pumps in these Yappa effector proteins. Those Yappa effectors disrupt whatever's going on in the cell so that you don't get packaging of those important components into the EV. The number of the EVs that are produced goes down, and those that are produced lack the cargo that will either directly kill the bacteria or initiate responses by those bystander immune cells. So we really think that this is a mechanism that contributes to the development of that non-inflammatory phase that's super important for the bacteria to colonize the host. Now we have outstanding questions here that we're still working on. We really want to know the mechanisms that are used by Yersinia pestis. And the important part here is, we can use this now, again, as a model to understand how EVs go through biogenesis. This is completely studied in the neutrophil world on how you actually get stuff into the EV, how it changes. And then ultimately, we want to get back to what we think is going on, is that these EVs actually contribute to developing that non-inflammatory environment. So I want to bring this back to my title. My title was A Plague on Your PMNs. And so neutrophils, we talked a little bit about some of the inflammatory responses that neutrophils contribute to. But of course, they're really famous for their ability to directly kill bacteria, right? Now you can do this by phagocytosis, by the production of reactive oxygen, etc. And so this is one of the reasons why they're first responders. They can come in, quickly respond, kill, and then recruit in other cells. If you look at every single one of these responses, we and others have now shown that this bacteria inhibits every single one of these responses. So Caitlin and Amanda like to call this the master manipulator of the neutrophil. And this is where the power comes in to understand the biology of neutrophils by using it. All right, so as I said, I highlighted Amanda and Caitlin who really drove all this work. This is my lab up here. Iman is a bioengineering undergrad. He's been working in the lab, actually worked with me as a high school student for a long time, who's been contributing to some of this work. We have a lot of collaborators highlighted in red here. I think I'll wrap it up because we're getting close to time here. And I appreciate you guys coming out this early after winter break. So thank you much. Yeah.
Then stayed in that time state, or they be then driven into that contra and
A great question. So we're doing that now. We're starting to do single cell sequencing on those to see if it's not just staying in the naive, but driving it to the M2. There's a lot of markers that we can look at for that. I think In many cases, what's happening is we're not getting polarization either way, so they're kind of staying in that naive state. But I think going either to staying in naive or going to M2 will be beneficial for the bacteria either way. Oh, I'm sorry.
So the next step with this really just to look at like how these YOP proteins gum up everything in a neutrophil, because it seems like the Yersinia is injecting these YOP proteins and it's just gumming up everything.
Yeah. So I didn't have time to tell you guys, but we take the same approach that Amanda took when she looked at LTB4 and added back all the YOPs to see what would happen. And so what you can see is that it actually takes 3 YOPs, YOP E, YOP K, and YOP H to inhibit it. But yes, we're super excited about this because we know that YOP H blocks calcium signaling. So how does calcium signaling contribute to the biogenesis of the E? What we're really interested in. YOP E is a Rho GTPase. So how does Rho signaling contribute to this? We are now investigating that. And then YOP K is a weird one. It blocks inflammasome activation, which has never been linked to EV production, but inflammasome can lead to cell death, and cell death does result in EV production. And so we're really interested in using this as tools to really get at the biology of how EVs are produced. And then we can go in the proteomics and say, are the antimicrobials in there because of calcium signaling? Or are the antimicrobials in there because of the gap, or the Rho signaling that's going on? And so how do those signaling lead to packaging of antimicrobials versus packaging of other signaling molecules versus we haven't even got into lipids and small RNAs yet, but we think that those are going to be differentially packaged also. So I think you're absolutely right. How's it gumming it up, and how does that allow us to better understand the biology, the cell biology of the neutrophil itself? Oh, yep. So potentially, I mean, I don't think we see a lot of change in the cell wall. One of the things that you might find interesting for pestis is gram negatives typically make LPS on their outer membrane, right? LPS is recognized by TLR4. It's a potent camp that stimulates immune responses. Turns out that pestis during evolution has changed its LPS by changing the number of lipid molecules, side chains on the lipid A part of LPS to not be recognized by TLR4. So there's the active subversion of the cells, but there's also the passive immune evasion properties that contribute to this. We haven't looked at how TLR signaling might contribute to it, but I guarantee it probably does. Yep.
Now that immune cells are highly migratory in that sense, have you thought of or have you looked into any potential influence on how well the neutrophils are still able to exhibit this kind of sentinel behavior?
Yeah.
Kind of dysregulation.
Yeah, there's 2 ways that we can look at that, right? So there's one that is what's going on inside the cell. And I will tell you, the longer you infect these cells with Yersinia, they'll start to round up. The YOP E actually, part of that Rho GTPase also screws up actin polymerization, right? And so you don't have calcium signaling to lead to metastasis. If you don't have actin polymerizing correctly, you're not going to have this motility. If you're not making LTB4, which is a chemoattractant, you're also not going to recruit in the other ones that aren't interacting with it. So I think the chemotaxis, the motility, and then maybe even more important than that, the phagocytosis that's going to be dependent on calcium signaling and actin polymerization to eat the bacteria are all getting disrupted. So you're getting disruption on the ones that are interacting, you're getting disruption on the bystanders, and this is all contributing to this ability to not contain the infection and lead to it. pneumonic plague is 100% fatal if you don't get treated. And that's true in the mouse model or in humans. And so it definitely is an uncontrolled disease without antibiotic treatment. All right, and take a look at the Center for Predictive Medicine if you guys are interested in infectious disease research. There's a lot of people over there that would benefit from bioengineering students that are interested in drug delivery and vaccine development. So, thank you again.
Google pages right after. I can try doing better.
Sorry. Oh. It'll be the whole time, yeah.
No, I'm saying that. And again, I appreciate it. It's always good to get over on this campus.
Anyone reaching out?
Yeah, yeah, point mine very, if I don't have anything, because there's a lot of people over there that are putting.
I was like, that one ran, make sure that I accurate.
Very.
Thank you. Thank you.